Once upon a time, there was a species named the circulatorians. They were thriving in the ecosystem that you call your veins. They were all quite similar looking, red and round, and they could all do similar things, like carrying oxygen all throughout their ecosystem, except for one. His name is Mike, and he's very special.
Mike is known as a sickle cell, and this is due to a very particularly interesting mutation. You may be wondering what a mutation is. Well, you're in luck. A mutation is the change of an organism's DNA sequence, and because of the change, it can affect how the organism functions. One of these changes is very obvious when looking at the cell as a whole. This particular mutation of Mike causes his shape to be drastically different when compared to other circulatorians.
His mutation causes his protein, or more specifically his hemoglobin, to be defective. Because of these defective hemoglobin within Mike's body, he lacks the ability to carry oxygen, altering Mike's structure and his appearance to more of a crescent or sickle shape compared to a normal-looking cell. Being born with this obvious difference caused Mike to be bullied and looked down upon his whole life, but if only they knew his true potential.
But how does this happen? How has such a drastic change occurred? A mistake is made in the process called transcription. In Mike's case, when he was born, something very special happened to the way his DNA is organized. You see, when a circulatorian reproduces, they make an exact copy of themselves to then divide. But when Mike was created from his identical copy and the DNA was split into two, a mistake occurred deep within the circulatorian that created him.
Within all circulatorians, there are more organisms working together. One of these organisms is often called the enzyme, but to the circulatorians, they are known as enzymites. Their main function is to speed things up. One enzymite named helicase has a goal of separating a DNA strand for easy access to get information. Another organism, named RNA polymerase, takes the information in order to start the process of translation. They both do this in a place called the nucleus. They both help the circulatorians to replicate their DNA as quickly as necessary. Another organism titled messenger RNA, or mRNA for short, collects that information to then be translated into something the other organisms can understand and use.
Another possible complication that may have occurred during Mike's creation would have been during translation. Translation is where the DNA strand is converted by mRNA, or also known as messenger RNA, into something that tRNA, or also known as transfer RNA, can understand. In the circulatorians world, they are known as the transcription team, with different people assigned to different roles. Their names are between DNA and different types of RNA, and they have this weird issue. They can only understand each other if what they are saying is the complete opposite. For some interesting reason, RNA also can't say certain things back to DNA, so it substitutes certain things as well. During this process, different letters are exchanged to different letters. The ACGT letters for a DNA strand need to be transferred to an ACGU for an RNA strand.
During this process, a mistake in translation can occur when the wrong letter is understood from the other. One specific example of this is between RNA and DNA. The other circulatorians in the sixth position would have the DNA letters GAG, which can be then converted into another group called the amines. The codons would be the letters GAG, while the anticodons would be the opposite, hence the name anti. The amines need to separate the letters into groups of 3 into where codons are. These are all made on a ribosome, where different proteins are made to build essential items. Along with this is someone named Peptide, who loves to bring things together. He usually helps connect these amines together by creating his special bond. After following all procedures, the specific letters make the amino acid glutamine, but sometimes it can be mistaken. When Mike was being created, his sixth sequence was GTG, which is valine.
This mistake seems to be a substitutional issue, as the letter A was substituted to the letter T. This can cause a completely different amino acid, which is what happened to Mike. If this error continues repeatedly, the continuation of this mistake led to a change in the genetic code responsible for producing hemoglobin, which is responsible for the oxygen in a cell, resulting in the unique sickle shape of Mike.
Our story continues with Mike facing challenges due to his distinctive shape. Other circulatorians often wondered why he was different. Despite the initial struggles, Mike discovered that his mutation provided an unexpected advantage. He was more resistant to a notorious villain named malaria. Malaria, a parasite transmitted through mosquito bites, posed a serious threat to the circulatorian community.
Mike's altered hemoglobin structure made it difficult for the malaria parasite to invade and thrive within his cells. The unique bonding properties of his hemoglobin hindered the parasite's ability to reproduce, showcasing the importance of genetic diversity in the face of environmental challenges. Malaria often depends on the oxygen in the red blood cell, but since Mike has less oxygen compared to other red blood cells, it is harder for malaria to live.
As malaria attempted to spread through the veins, Mike became a hero among the circulatorians. His resilience and ability to fend off the villainous parasite highlighted the significance of genetic adaptations in the survival of species.
In the end, the circulatorians learned to appreciate Mike's unique mutation, recognizing that diversity within their community played a crucial role in overcoming threats. The story of Mike, the sickle cell, became a valuable lesson in biology and genetics for all the young circulatorians, emphasizing the importance of adaptation and embracing differences for the well-being of their community.